Hello everyone and welcome back to PhytoPathoVerse. I'm Malini, molecular plant pathologist with a motto to make plant pathology easy. So after a short pause in this series of GN Agrios Made Easy, I received request to continue it. So here we are with chapter 3, that is Pathogenicity. How do plant pathogens cause disease? But before diving into chapter 3, let's quickly recap what we have learned in chapter 2. First, we have seen disease cycle, then effects of pathogen on various plant physiological aspects. Okay. The first one is photosynthesis, then photorespiration, respiration, transpiration, translocation, plant growth and reproduction. Now let's see what we have in our chapter 3. We will start with the word pathogenicity. That means the ability of a pathogen to cause disease, right? Now what are pathogens? We have seen examples of which in chapter 2. Okay. Pathogens are such as fungi, bacteria, nematode, molecules, virus, viroids, right? Here we will see pathogenicity is a rare phenomenon. Okay, and all the pathogens which we see, which are available, they all don't have the ability to cause disease, right? Now, there are estimated to be 1 to 2 million species of fungi, but only about 10,000 have been linked to plant diseases, right? Similarly, we will see there are about 30,000 bacterial species, but only around 250 are plant pathogenic. Again, we will see a small fraction of nematodes, viruses and plants are phytopathogens, right? So, what are the properties that this small fraction they have that other don't, right? That we will see in the following section, okay? So, to colonize plants, pathogen must accomplish a series of crucial steps. So, what are these steps? What are the methods or process due to which the specific number of pathogens they are pathogenic? Means the microorganisms means 10,000 fungi they have the ability to cause disease. The rest 1 million species they don't. So, they must follow this series of crucial steps. First is recognition of the host. Second, growth and movement towards it. Yes. Next, attachment to the surface. Surface means host plant, right? Passive entry or active penetration. Okay, means penetration. Okay. Next, evasion of preformed and recognition induced defenses. Recognition. Next step, acquisition of nutrients through hostoria. Okay. Next, growth and proliferation within the host. Okay. Next, reproduction, which is necessary step, and that's it. Dissemination means after doing their work, they need to be spread to new host, right? Now, the failure to successfully complete any of these steps will lead to the extinction of this lineage of pathogen. So, this many series of crucial steps need to be followed, and failure to any one of these steps will lead to the extinction of this lineage of pathogens, right? Now here we will get to see one interesting thing, that the phyla that include the major group of pathogens all emerged well before the evolution of vascular plants, and it was around 300 million years ago. Whereas the major phase of angiosperm diversification was around 100 million years ago. So the pathogens were in this environment way longer than the angiosperm. Okay. So with this, we can conclude that only a few species initially evolved the valuable ability to cause disease on plants, right? It is also clear that all extant plant species are resistant to all but a few dozen pathogen species. Also, we can conclude that there has been an evolutionary arm race between the host means the plants, and the pathogens, right, leading to the current interaction. Alright, whereby most pathogens can only infect a few plant species. The nature of the evolutionary innovations that enabled pathogenicity and host specificity constitutes one of the core fascinations of plant pathology. Now, there has been huge progress in our understanding of pathogenicity in the last 20 years. Why? Because of the large part which played in application of genomic data. Okay. Here we will see using the tools of functional genomics means the ability to study the role of individual genes during infection. It has become clear that specific pathogens often have specific genes that enable pathogenicity on specific host, right? Okay. Now, a common theme has emerged. A successful plant pathogens must be able to invade a plant while combating its immune response. See, when any attacker or some external foreign particle or person is trying to harm us, what do? We have defense mechanism, right? We try to fight them back. So that is the immune response. So when any pathogen is trying to harm the plant, so they automatically activate their defense mechanism. Now, a successful pathogen, what they are telling is that a successful plant pathogen must be able to invade a plant while combating means fighting the host immune response. Okay. This is achieved by different groups of pathogens in different ways and at different times during infection. Okay. So, the study of pathogenicity is not only scientifically interesting but is also interestingly being applied directly to improve the control of diseases. Okay. Here in the left hand side we will see a definition of pathogenicity. What they are saying, let's see once, is that there was much of the confusion and controversy in plant pathology is caused by the inconsistent definition of key terms, right? So here we will see the how we can define pathogenicity. So it is the ability of any isolate of pathogenic species to cause disease on any line of a host plant species. If only some isolates of a pathogen can cause disease on a specific host genotype, such isolates are described as virulent. Okay. Isolates that cannot cause disease are avirulent. One more term we will get to see here is aggression. What is it? Aggression refers to the degree of disease. More aggressive isolates cause more severe and damaging disease, What are the terms that we saw here? First is pathogenicity. We have already discussed ability of pathogenic species to cause disease. Next, there were virulent and avirulent. The pathogen that can cause disease, they are virulent, and which they cannot cause disease, they are avirulent. Now, what is aggression? The degree of disease. Like for example, two friends are fighting, so they both have the ability to fight. That means the both friends are virulent pathogen. Okay. Now, and between two friends, one will win and one will lose. The one who will win, he or she is more aggressive. Okay. So even after having the ability to cause disease, some pathogen, they have more aggression than the other. So they are called, the degree refers to the degree of disease. More aggressive isolates have more severe and damaging diseases. I hope it's clear. Okay. Let's see what do we have here. Different plant infection strategies and pathogen lifestyles. Traditionally, plant pathogen infection strategies and lifestyles were classified into two divisions, biotrophy and necrotrophy. Based on what? Based on their mode of nutrient acquisition from the host. So what biotrophy they are referring? It refers to the extraction of nutrients from living plant cell. Just remember this point. Okay. Biotrophy means they extract nutrients from living plant cells, whereas necrotrophy, what they do, they kill the plant cells. Okay. They kill the plant cell and then they extract nutrients from the dead and dying cells. Okay. Biotrophy, they extract nutrients from living cells, whereas necrotrophy, what they do, they kill the plant cells and then they feed off from the dead and dying cells. Okay. For now, just remember that. Now we will see how do they penetrate in the plant cell. Okay. The intact healthy plant is a community of cells built in a fortress fashion. Fortress like fashion. Now we have seen forts. Forts means the palace or the building which was used by kings or emperors to protect from enemies, right? So this plant cells, they have all this defense. See, plant cell wall is consist of what? Middle lamella, primary cell wall, secondary cell wall, plasma membrane, all these layers. So they protect them from penetration. Okay. Now let's see, plant cell consists of cell wall, cell membrane, cytoplasm, which contains of nucleus and various cell organelles. Okay. What are the cell organelles that we might have studied in our biology in schools, right? So what they do, they have a series and layers of protection. So what is the best way for them? For them means for the pathogen to penetrate. So what they do, in the aerial parts of plants, there are breaches on the exterior surface. So what are the breaches? The most obvious are the stomata. Stomata we have all heard, right? Which open to allow the plant to exchange gases. Now, some pathogen, they enter through stomata. If not stomata, they also have lenticels. Lenticels are what? The pores found in woody stems and roots. If not that, they have hydrathodes. Hydrathodes are pores found in extremities of leaves through which excess water is extruded, right? Along with these openings, the other openings are formed when they are wounded. Wounded means they are harmed. Okay. See, when the plants are wounded by grazing, mowing, pruning or other physical damage. Any physical damage, any wound, there will be a breakage in the protection layer. So, the pathogen can penetrate. Okay. Now, a universal feature of a successful pathogen is that the infectious propagule, whether it be a fungal or oomycete spore, a growing mycelium, a virion or a bacterium, must be able to penetrate the exterior surface of the plant, be it any pathogen. These are the example. Any pathogen, the first thing that they need to be done is penetration. It can be any natural opening like lenticel, stomata, hydrathorax or any wounding, or by any other method like vector, okay, but they need to penetrate. The strategies used to penetrate plants, it can be divided into 3 categories. What are those? One is direct penetration of the epidermis, second is entry through stomata or any other natural openings, and the third one is vector organisms. Okay. First, we will see direct penetration of the epidermis. The epidermis represents a formidable barrier for the invading pathogens. Formidable means which forbids the pathogen for entry. Okay. Now, to penetrate a plant surface, pathogen propagules may first adhere to it. Now, what is this adhere? Adhere means to stick firmly. Stick means everybody knows, right? So, now after the contact is established means they have adhered to the epidermal layer. So, after the contact is established, the diameter of the tip of the hypha in contact with the host increases and forms a flattened tube-like structure called aprasorium. Now, the aprasorium then will develop into a penetration peg, okay, which advances into and through the cuticle cell wall. Here some examples are given like Sortyromyces, Colletotrichum, Geimanomyces, Pyricularia magnaporthi. Okay. So, in these cases, penetration of the plant cell takes place only if melanin accumulates in the aprasorial cell wall. What is melanin? It is a polymeric black pigment. So, whenever melanin will be present, it helps in the penetration of the given pathogens. Okay. See, in this diagram, it will be a little more clear. First, this is the conidium. Means we in this case we can say this is the infection propagule. Okay. So, first the conidium, it will get attached to the host surface. Now, the germ tube will start growing and then it will form a bulbous like shape called aprasorium, and it will develop to penetration peg, which will help in penetration into the host cell. Then primary invasive hyphae, it will start spreading. Okay, the tissue will colonize and then it will disseminate. So, from this it will erupt and the conidia will spread in the environment, which will help them target a new fresh healthy plant. Okay. Now, the second mode of entry was through natural opening. So, rather than the penetrating through epidermis, a small number of pathogen groups have specialized to penetrate through the stomata. So, they are clever enough, So, they simply use the natural openings and penetrate. So, this might seem to be a rather obvious strategy, and surprisingly few pathogens have mastered the art. So the third mode of entry is through vectors. Viruses and viroids require vector organism to transfer from the plants and penetrate the host. Now, see here, just remember about viruses, not viroids, okay? Because viruses can be transmitted through vectors like insect vector, but for viroids, they only require and they only transmit through mechanical transmission, okay? Okay, so viruses are vectored by sap-sucking insects, chewing insects, and plasmodiphoroid like Polymixa betae. Okay. Now the nutrient sources for penetration. Until the pathogen can form a hosterium or reach the rich resources of the apoplast and the vascular tissue, it must rely solely on nutrients stored in the spore. What happens in case of fungi? The carbohydrate is stored as glycogen. This question comes in competitive exams. So remember, in case of fungi, it is stored as in the form of glycogen. In case of oomycete, the carbohydrate is stored in the form of mycolaminarin. Also, similarly, for bacteria, the carbohydrate is stored as beta hydroxybutyrate. Okay, all these questions comes in competitive exams. Okay, so the invasion and initial colonization. We will see what happens in after initial colonization. Hostoria and related structures. Okay, so hostoria represent one of the most remarkable structures found in plant science. They are found in many fungal groups: Puccineals, Eustilaginales, Erysipelas, Cholestrotrichum, Pyricularia, and others. All oomycete pathogen groups as well as all the parasitic plants. They are also found in many mutualistic symbiotic relationships such as arbuscular mycorrhiza. Now, the key properties of the hostoria is that it enables the pathogen to extract nutrients from host cell while maintaining it in a living and fully functioning state. Okay, detoxification of plant defensive compounds. Now, we have discussed that plant, they have defensive mechanism, right? If somebody will attack you, obviously you will attack back, right? That is their defensive mechanism. So how pathogen detoxify the plant defensive compounds? First, we will see what are these defensive compounds. Now, plants biosynthesize a vast array of small molecular weight compounds known collectively as secondary metabolites. Now, many of these have roles in deterring herbivores. Now, deterring herbivores, how they help in defense mechanism? Because herbivores are one of the vectors responsible for transmitting a virus or any other pathogen, right? So, if the secondary metabolites can deter means they stop herbivores, the insects. So, the what? If the mode of transmission will get stopped, so there will be no way of entering virus into the plant cell, okay? But a few appear to have the prime function of inhibiting microbial pathogens. Now, look, plants biosynthesize as many as secondary metabolites, but all are not function for inhibiting microbial pathogen. Only few work in that way, okay? Now, it is very difficult to disentangle the role of individual plant secondary metabolites in plant pathogen interaction, except by showing that pathogens with altered capabilities to detoxify these compounds have altered pathogenic properties. So it is very difficult which secondary metabolites is responsible for deterring herbivores or any other defensive mechanism. So how we identify them? Simply by showing that any presence of any secondary metabolite, and if it stops the pathogen attack, or they alter the capability of the pathogen, so they consider it as active against the specific pathogen. That's it. Now, there are two well-studied defensive secondary metabolites. One is saponins, due to their soapy nature. Saponins are the secondary metabolite we see in vegetable plants normally. Okay. Now, along with saponin, there are tomatin, which we get to see that is present in tomatoes. Okay. Okay. Now, obtaining nutrients from plant cell walls. Everybody is clever. So, plant cell walls are composed primarily of carbon polymers, the most abundant being cellulose, followed by pectin and xylem. Okay. So, these polymers serve as potential source of carbon nutrients for whom? For the pathogen. They obtain nutrient from their host, right? From the cell wall, from the apoplastic layer, from the space between them. So they are very clever as well. Although this requires their cleavage into monomers that can enter the pathogen cell to be used in primary metabolism. Now, both bacteria and fungi are known to produce a plethora of different enzymes which cooperate to deconstruct the plant cell wall. Okay. That's it. They have given some examples how they do that. Okay. We are keeping it simple. So, moving directly to invasion of the xylem. Now, vascular wilt pathogen have evolved specific mechanism for invasion of the plant xylem. Now, few of the pathogen, they obtain nutrients from plant cell walls, whereas the other part, few of them, they invade xylem vessel. There are two vascular bundles, xylem and phloem. Few, they attack xylem vessel, and we heard of vascular wilt or fusarium wilt. Vascular wilt, they are mainly caused by fusarium oxysporum, but there are more species, okay, but mainly through fusarium. Similarly, we have also heard the pathogen which infect in phloem vessels, okay, such as the phytoplasma. They mainly target the phloem vessel. Why? Because phloem is responsible for translocating food for the plants, right? Okay. Now, pathogens that seek to cause cell death. An important class of pathogens has the ability to invade the apoplastic space by direct penetration between epidermal cell walls, even though they can often be observed to enter via wounds or natural openings. Having gained entry to the leaf, their strategy lies in direct contrast to the stealthy apoplastic pathogen described above. Yes, we have seen this stealthy apoplastic pathogen that remains in between the spaces. So, instead of seeking to remain camouflaged, virulence in this group is due in large part of the secretion of a diverse family of effectors that induce chlorosis and necrosis of the plant tissue. Okay, so this type of pathogen, they kill the plant cells, and the pathogen which are responsible for killing the plant cells, and after killing the plant cell, they feed on that dead and decaying cells. So that type of pathogen are called necrotrophs, right? We have seen it sometime before. They are necrotrophs. Okay. Some of the examples of those, they are present in Pleosporales family. All right. Okay. Now, do you know how they kill the cells of the plants? So they do it with the help of some toxins. They release some toxins due to which the plant cells, they die. One of such example of toxin is Victorin or HV toxin. Okay. And this HV toxin, Victorin, it's important. Remember this toxin name because it is the first host specific plant toxin. Okay, it comes in exams and it is produced by fungus Bipolaris victori. Okay, one more example is T toxin, and it is produced by the race T of Bipolaris maidis. Just remember this much in this. These toxins are responsible and they help the pathogen to kill the plant cells, and after the cell is dead, that necrotrophic pathogen, they feed on that dead and decaying matters. Okay. Next that we have is crown gall disease caused by Agrobacterium. Yes, Agrobacterium tumefaciens, they cause crown gall disease. Nothing to read more. If you want to know it deeply, please refer to this section. But in a simpler way, just remember Agrobacterium tumefaciens, they cause crown gall disease. Okay. Okay. Next, growth regulators in plant disease. Now there are various growth regulators we have heard of, which are auxins, gibberellins, cytokinins, ethylene. So they all are responsible for causing diseases, or they help the pathogen. So what they do, we will see. Plant growth is regulated by a small number of groups of naturally occurring compounds that act as hormones and are generally called as growth regulators. The most important growth regulators are auxins, gibberellins, cytokinins and other compounds such as ethylene and growth inhibitors. Okay. So yeah, these are growth regulators. Okay. Now, these growth regulators act in very small concentration, and even slight deviation from the normal concentration may bring about strikingly different plant growth patterns. Like see, we have heard of Bakani disease of rice. Yes. So, Bakani disease of rice is caused by what? This gibberellin. Okay. And this disease is also called fully seedling. From that disease, this gibberellic acid was first discovered. Now, the plant growth hormones, they have, they act in very small concentration. So, in that case, in Bakani disease, the concentration is deviated, and that's why the seedlings, they have grown taller than the normal height of rice, right? So any small concentration, any slight deviation from the normal concentration may bring different plant growth patterns. We will start with auxins. Auxin. The auxin occurring naturally in plants is indole 3-acetic acid, which we normally called IAA, and produced continually in growing plant tissues.
IAA moves rapidly from the young green tissues to older tissues but is degraded constantly by the enzyme IAA oxidase, which explains the low concentration of the auxin. We will see here the effect of IAA on the plants are numerous. Numerous means more than one. There are numbers of effects of IAA on plants. What are those? It is required for cell elongation and differentiation. Okay. And absorption of IAA to the cell membrane also affects its permeability. Okay. Okay. Coming to gibberellin, gibberellin are normal constituents of green plants and are also produced by several microorganisms. Gibberellins were first isolated from the fungus Gibberella fusiculae, which causes fully seedling of rice, just mentioned few minutes before. Okay, these fully seedlings are also called Bakane disease of rice, right? So the best known gibberellin is gibberellic acid. Okay. Next we will see cytokinin. Right. Cytokinin. Cytokinins are potent growth factors necessary for plant cell growth and differentiation. In addition, they inhibit the breakdown of proteins and nucleic acid, causing inhibition of senescence, and they have the capacity to direct the flow of amino acid and other nutrients through the plant toward the point of high cytokinin concentration. Okay, so cytokinin occurs in very small concentrations in green plants, in seeds and in the sap stream. The first compound with cytokinin activity to be identified as kinetin, okay, which however was isolated from herring sperm DNA and does not occur naturally in plants. Okay. Next, ethylene, produced naturally by plants. Ethylene exerts a variety of effects on plants, including chlorosis. Chlorosis means yellowing, deviation of chlorophyll, okay, leaf abscission, epinasty, stimulation of adventitious roots, and fruit ripening. So ethylene also cause increased permeability of cell membranes, which is common effect of infections. Okay. Next we will see late stage infection. Here, although there is no consistent and clear-cut differentiation between early stage infection and late stage infection, but it's better to keep them separate. Okay. So, in late stage infection, pathogens are seeking to acquire sufficient nutrients to complete their cycles and ensure they have to resource to initiate a new infectious cycle. Okay, so they have completed whatever they needed to do. Now their new motivation is to start a new cycle. So the completion of pathogen life cycle often involves toxin release, both to kill the cells and to inhibit secondary invaders. Now we will get to see little bit about toxins, right? Now toxins can be divided into host specific and general. Okay, non-specific toxins primarily operate in late infection cycle. Why? The host specific toxins, they are specific to that host. Okay, now look, there are difference between host specific and non-host specific toxins, and many questions come from the examples of host specific and non-host specific toxins. Next, we also have mycotoxins. So mycotoxins are secondary metabolites produced by fungi such as Fusarium, Alternaria, Aspergillus or Penicillium. while the name mycotoxins generally indicates fungal compounds that are harmful to humans and animals. And whenever they will be toxic to plants, they will be called as phytotoxins. So one of the oldest known phytotoxin is fusaric acid, Fa, which is produced by Fusarium species. So fusaric acid was the first fungal phytotoxin isolated from an infected host plant and displays strong toxicity to plant but only moderate toxicity to animal cells. Okay, remember this. Next we will see tap toxin. Now tap toxin is produced by the bacterium Pseudomonas syringae pathogenicity, which causes the wildfire disease of tobacco. Remember this. Question come both. This tap toxin is produced by which pathogen? That is Pseudomonas syringae pathogenicity, and it is also called wildfire disease of tobacco. By stains of PathoVerse occurring on other hosts such as bean and soybean, and by other pathogens of Pseudomonas syringae such as those occurring in oats, maize and coffee. This toxin producing strains cause necrotic spots on leaves. That's it. These are the symptoms, okay, of this toxin. Now we will jump directly to the terminal phase of pathogenicity. While doing these steps, if we follow it precisely, we will see so many factors are responsible for a pathogen to cause disease and for a plant to defend them. Okay. Now we will see the terminal phase. So the final phase of pathogenicity can be defined as the production of symptoms and the formation of pathogen propagules, which are capable of transmitting the disease to the next generation of host, right? Now production of infectious propagules. What do we see here? Each class of pathogen such as fungi, bacteria, oomycetes, viruses, nematodes and parasitic plants, they produce different types of infectious propagules, often more than one type. To be a successful pathogen, the propagule must be able to move to a new susceptible host. If they will not move to the next susceptible host, then their lineage will stop there, right? So they need to move. So some pathogens produce light and dry spores so that they can be blown by the wind. With the help of wind, the spores can disseminate. Others produce heavier spores that are translocated by splashing of rainfall as it hits the crop, whereas others can move under their own power using flagella. Where do we see flagella? Mainly in bacteria, right? But there are also some viruses, there are also some fungi or oomycete where we get to see flagella, particularly in water adhering to soil particles. Yet others are moved by vector organism. Which type of pathogens are moved by vector organism? They are mainly viruses, right? Now, not just viruses by insect, but also by humans and other animals and on farm machinery. So this vector means not only insect vectors. When we called vector, that means it includes any vector. One can be insect vectors, like yes, majority of the vectors are insect, but they are also humans or animals or any farm machineries, right? Finally, some pathogens adhere to the seed surface to penetrate within seeds and other plant materials and resume their pathogenic lifestyle when the plant starts to regrow. Okay. See here in the terminal phase we have seen what is the terminal phase? The production of symptoms, right? So we will now see symptom production. The macroscopic and microscopic appearance of symptoms on the diseased plant is not only the visual manifestation of infection but also remains the primary means of diagnosis. Yes, in some diseases, the symptomology is dominated by the sporulation of the pathogen. Powdery mildew pathogens produce abundant strings of conidia on the surface of leaf directly from the superficial mycelium. We get to observe powdery mildew very prominently on top of the leaves. Okay, they have this powdery like substance. And rust pathogens, downy mildew, sea woolworm, venturia also sporulate profusely on the leaf surface, generating macroscopic symptoms. Right. So these are macroscopic symptoms that we get to see. They are not always internal. We have external, internal signs, symptoms, so many means of diagnosis. Okay. Many foliar pathogens cause chlorosis and necrosis that we have already seen. The photosynthetic machinery uses light to generate an electrochemical photon gradient, which in turn drives the biosynthesis of sugar and other biomolecules. In this highly regulated process, any part in this highly regulated process, any perturbation by the pathogen can result in the release of ROS, that is reactive oxygen species, that can damage all cellular components including chlorophyll. And if the chlorophyll will be damaged, we will get to see chlorosis, means yellowing, right? Okay, this is the starting point of yellowing, chlorosis. Okay, that is so often a primary symptom of infection. Now, if exposed to more sunlight, the unquenched photochemical activity can further damage the leaf and lead to cell death and necrosis. In addition, perception of the damage by the plants induces the pathways of programmed cell death (PCD). Root, crown and stem pathways typically block the vascular tissues, leading to wilting and leaf drop. The result from the perspective of the plant is disease. The reduction in photosynthetic output, which has the potential to reduce biomass accumulation and seed yield and quality. The result from the perspective of pathogen is the production of viable propagules ready for next infection cycle. Now, at last, we need to see what is the result that we should observe in perspection of plant and with the perspective of pathogen. Let's read it again. The result from the perspective of plant is disease. Okay, and the result from the perspective of pathogen is the production of viable propagules ready for the next infectious cycle. All right, so that's it for today, and I hope it was not that typical or critical or complex, because I always try to keep it simple. And yes, there are more concepts and we can go deep and learn each and every bit of each and every word or terms which are related. But the series are intended to make it simple, not complex. So, okay, we will be back with the next chapter soon. Till then, keep learning, keep growing. See ya.